A warm welcome to all of you. Today we are discussing cell cycle and diagrams.
What is cell cycle? Divide, grow and redivide. That is cell cycle. Let us check the diagram. This is the diagram of cell cycle that consists of two main phases, M phase and interphase. M phase means either mitosis or meiosis that consists of two stages, karyokinesis and cytokinesis. Then the next phase, that is interphase. Interphase consists of G1, S and G2 phase. The details we already learned in the last videos, right? Yes. Here you can see is after M phase, daughter cells will form. Here we are discussing about mitotic phase. Yes, two daughter cells will form. Yes, these two daughter cells will either enter into the next cell division or it will not enter into the next cell division. Example, nerve cell. Once it formed, it will not divide again. Then the interphase is not there for the nerve cell. But in some cases, that is the daughter cells will enter into the next cell cycle. That is, it will enter into the interphase. Is meristematic cells, rapidly dividing cells, that is meristematic cells that you know. Yes, that type of cells enter into the interphase, then that will undergo M phase. Like that cell cycle will continue in meristematic region. But in some cases, example nerve cell, there is no cell division. Then yes, liver cells. Liver cells is whether it is necessary, it will divide. Then based on the cells, yes, the cell cycle will continue. Cell cycle will continue, yes, in meristematic region or in growing region. Can the cell cycle go on endlessly? Yes, what is your opinion? No. At some places it stops permanently. Example, brain and nerve cells. Once it formed, it will not divide again. In some places, it stops temporarily. Can you say examples? Yes, liver cells. Here you can see these are the liver cells. Yes, whenever it required, it will divide again. Yes, it will stop. The cell division can stop temporarily. Then whenever it requires, it will enter into the cell cycle. That cell will enter into the cell cycle. That is in the case of liver cell. The next point, in some places cell cycle go on till it is needed. Surface of the skin. Here you can see epithelial cells of the skin. Yes, the cell cycle go on till it is needed. Then the next point, in plants, the growing points, that is meristematic regions of the plants. Here you can see in this picture, yes, these are the meristematic regions of the plants, apical meristem, intercalary meristem, lateral meristem. These are the different meristems present in plants. These meristematic regions, cell will continuously divide. Rapidly dividing cells you can see in the meristematic regions. In these regions, cell cycle will continue. Uncontrolled non-stop cell cycles may lead to tumors that may or may not be cancerous. Yes, non-stop cell cycles are very dangerous, that may be cancerous. Cell production and cell death. We can check in different age groups. In children, the production of new cells is more than the dying cells. In adults, new cells is equal to dying cells. Then the number of dying cells replaced by new cells. In old people, new cells less than dying cells. Homologous chromosomes. What are homologous chromosomes? A pair of corresponding chromosomes of the same shape and size, one from each parent. That is called homologous chromosomes. Here you can see this is a human karyotype. There are 23 pairs of chromosomes are there in human beings, right? Yes. If you check the first pair, you can see that is in same shape and size, one from each parent. That means one chromosome from each pair is coming from mother. That is called maternal chromosome. Then the other chromosome from that pair coming from father. That is called is paternal chromosome. Then maternal and paternal chromosomes are there in each pair. And if you check the human karyotype, 22 pairs of autosomes are there plus one pair of sex chromosomes are there. Then in male, that is X and Y, the sex chromosomes are X and Y, and in female, that is X and X. Meiosis. Meiosis is another type of cell division occurring both plants and animals. Where it will occur? It will occur in reproductive organs of plants and animals. They are also known as reduction division. Reduction division producing sex cells or gametes that produce in or produced from sex organs. The most significant aspect of meiosis is that the number of chromosomes in sex cells is halved. Right? That means, suppose an organism is having 16 chromosomes in their body cells, then what is the number of chromosomes in that organism's sex cells? That is 8. 8 chromosomes will be there in their sex cells. Here's one more example. In humans, 23 pairs of homologous chromosomes are there. That is diploid or 2n. We can say 2n. Diploid in nature. Then, in that, one member of each pair are passed on to the sex cells. You know that each pair having his maternal and paternal chromosomes are there. Yes, these chromosomes, one member, either paternal or maternal, are passed on to the sex cells. Then what will be the number of chromosomes in sex cells of human beings? 23 chromosomes. That is haploid in nature, that we will represent N. In humans, meiosis takes place in reproductive organs. Yes, what are the reproductive organs of humans? In male, that is testis, and in female, that is ovary, to produce sperms and ova. Sperms from testis and ova from ovary. In flowering plants, meiosis takes place in the anthers and the ovary to produce pollen grains and ovules. The same way, yes, you know that anthers are the male reproductive organ of flowering plants that produces pollen grains, and ovary is the female reproductive organ of flowering plant that produces ovules. Significance of meiosis. The another name of meiosis, that is reduction division. Chromosome number is halved in gametes so that on fertilization, normal number is restored. The number of chromosomes in sex cells or gametes is haploid in nature so that on fertilization, normal number or diploid number of chromosomes will be restored. Body cell is diploid, but the sex cells or gametes are haploid. Then the next point, it provides mixing up of genes. These two are the main important significance of meiosis. First one, chromosome number is halved in gametes so that on fertilization normal number is restored. Then it provides mixing up of genes. What is mixing up of genes? The maternal and paternal chromosomes get mixed up as they separate during first reduction division from the homologous pairs. Here you can see these two are homologous pair of chromosomes. Then that will mixed up at the time of first reduction division. Yes, get mixed up. The genetic material get mixed up. Here you can see chromatids from same chromosome is called sister chromatids. Then chromatids from different chromosomes, that is non-sister chromatids. Then at time of crossing over, the maternal and paternal chromosomes are separating. The chromatid material very often get exchanged between the two members of a homologous pair, resulting in genetic recombination. Here you can see. Yes. Then the point of contact between two non-sister chromatids during crossing over, the point of contact we call it as chiasma. Chiasma formation will occur during crossing over. What are the differences between mitosis and meiosis? Mitosis occurs in body cells. Meiosis occurs in reproductive cells. Mitosis provides for growth and development, but meiosis only for gamete formation. Then the next point, mitosis occurs continuously throughout life, but meiosis occurs only reproductively active age. Mitosis, how many daughter cells are produced? 2 daughter cells produced during mitosis, 4 daughter cells produced during meiosis. A full set of chromosomes is passed on to each daughter cell in the case of mitosis, that is diploid in nature. That means parent cell and daughter cells have same number of chromosomes. Both are diploid in nature. But in the case of meiosis, only half the number of chromosomes are passed on to the daughter cell. In the case of meiosis, daughter cells are haploid. Half the number of chromosomes you can see, but in parent cell, that is diploid. Parent cells are diploid, but after meiosis, the daughter cells will be haploid. The next point, a single nuclear division after chromosome duplication. This nuclear division you can see only in one time. But in the case of meiosis, 2 nuclear divisions. Karyokinesis occur 2 times. Nuclear division means karyokinesis. It occurs 2 times after chromosome duplication. But in the case of mitosis, is how many nuclear division? Single nuclear division. 1 time. nucleus will be divided. The next point, daughter cells are identical. Both S2 daughter cells will be formed after mitosis. These two daughter cells are identical, exact copy. But in the case of meiosis, daughter cells are having genetic variations. That means you know that crossing over occurs during meiosis, then four daughter cells are different from each other. Then genetic variations you can see in daughter cells of S after meiosis. Diagrams and exam-based questions. The diagrams of different stages of mitosis are very important for examination. What are the different stages of mitosis? Prophase, metaphase, anaphase and telophase. These are very important for labeling, identification and drawing. Then is before mitosis, which phase will be there? That is interphase. That also you have to learn. Crossing over diagram is very important for examination. This diagram is for identification. Then you have to know the labelings, sister chromatids, non-sister chromatids. Then you can label homologous chromosomes, recombinant chromatids, chiasma. This all you can label. Some structure-based questions we are discussing here. First question, the diagram given below represents a stage during mitotic cell division in an animal cell. A. Identify the stage and give a reason to support your answer. You can see here is which stage of mitosis is this? This is anaphase. How to identify this? You can see here sister chromatids move towards the pole. That's why this stage is anaphase. Name the parts labeled 1, 2 and 3. 1 is centriole, 2 spindle fibers, 3 chromatids. What is the chromosome number of the cell? Yes, you have to check the chromatids. How many chromatids are there? 1, 2, 3, 4, and 4. The number of chromosomes in this cell is 4. Draw a neat label diagram of the cell as it would appear in the next stage. What will be the next stage? Telophase. Is you have to draw the diagram also. Just check out the answers. Here you can check the answers. Is in telophase diagram. You can label the other parts also. Centrosome you can label. Is nuclear envelope or nuclear membrane. You can label nucleolus. Then chromatin network also you can label, because you know that in telophase stage chromatids decondense to form chromatin network. Identify the stage and give reason to support your answer. How to identify the stage? Yes, you have to check whether this diagram is prophase, metaphase, anaphase or telophase. Yes, all of you know that metaphase means chromosomes arranged in equatorial plane. Here it is not like that. But in anaphase, chromosomes are, yes, chromatids move towards the pole. You can't see that one here, right? Then in telophase stage, two nucleus, you can see right? Then that also not here. Then the only possibility that is prophase. What are the special features of prophase? Chromosomes become distinct. Spindle fibers appear between daughter centrioles. Nucleolus and nuclear membrane disappear. Here you can see these all features, right? Chromosomes become distinct. Two chromatids, sister chromatids you can see in this chromosomes. Then spindle fibers appear between centrioles. Between daughter centrioles you can see spindle fibers. Then nucleolus and nuclear membrane disappear. Nuclear membrane starts to disappear. Then nucleolus also disappear. Then the stage is prophase. Identify the diagram and name the stage prior to this stage. Yes, which diagram is this? Here you can see chromosomes are arranged in equatorial plane. Then that is metaphase. Name the stage prior to this stage. What is the meaning of that? Prior means is just before the stage. Which stage is that? Prophase. Identify the diagram and label the parts. Yes, which phase is this? How to identify this diagram? Here you can see sister chromatids move towards the pole. Then this phase is anaphase. Then label the parts. First one, aster. Then second one, spindle fibers. Third one, chromatid. Given below is a schematic diagram of a portion of DNA. How many strands are shown in the diagram? Here you can see 2 DNA strands are there. The answer is 2. How many nucleotides have been shown in each strand? Each strand you can see 2 nucleotides. Yes, this is one nucleotide. This is another nucleotide. Then each strand you can see 2 nucleotides. Name the parts labeled 1, 2, 3, 4 and 5. 1 is phosphate, 2 sugar, then 3 nitrogenous base, 4 hydrogen bond, 5 nitrogenous base. Name the DNA units constituted by the parts collectively. Yes, that DNA units. Together we call it as nucleotide. Let us check. Identify the different stages of mitosis. In this picture, you can see the different stages of mitosis in plant cells. Yes, we can go through the different stages. First, we can find out prophase. Yes, what are the special features of prophase? Chromosomes are distinct. There is no nucleolus, no nuclear membrane. Yes, this is prophase stage. Other prophase cells are there that you can find out. I will circle only one prophase stage. The rest all you can find out. Then the next stage of mitosis, that is metaphase. Chromosomes are arranged on equatorial plane. Here you can see this is metaphase stage. Then the next stage of mitosis, that is anaphase. Yes, chromatids move towards the pole. So many anaphase stages are here. You can find out that. Then the last stage of mitosis, that is telophase. Telophase stage is there is no cytokinesis occur, but karyokinesis, two nucleus will be formed in one cell, that you can find out. In that way, you can find out telophase stage. Then nuclear membrane and nucleolus starts to appear. There is no distinct chromosome. Chromosomes decondense to form chromatin fibers. Here you can see this is telophase stage. Two nucleus in single cell, right? Karyokinesis completed, but cytokinesis not completed, right? This is telophase stage. Then other telophase stage you can find out. Then how to find out interphase? In interphase stage, nucleolus and nuclear membrane will be there. Then chromatin network instead of chromosome. Then that will be interphase. You can see here is this is interphase, right? Nucleolus, nuclear membrane, you can see. This is interphase stage. Thank you, children. Have a good day. If you like my channel, please like, share and subscribe and press the bell icon. Thank you.